Oh,
Rather than crossing my fingers and hoping that the virus train it.
you can't see me. So it's my extreme pleasure and honor to introduce Jennifer Listgarten with a top secret intro that she's not ready for. So Jennifer works in bioengineering and she's faculty in CS here. And I just saw that you're on the steering committee for BEAR. Is that correct? So, okay, cool. So Jennifer, formally, if you read about her, she works on protein engineering and she uses ML for protein engineering. But not only is she one of the pioneers of this area, I'm happy to say that Jennifer personally is actually a huge inspiration for the semester-long program. So I'm completely dead serious. Years back, work that Jennifer and one of her PhD students, Chloe Su, did on they did work on protein engineering with ML, but not only that, they both taught me about AlphaFold and the fact that AlphaFold does out of distribution prediction. You train it on a heavily human biased set of proteins and protein foldings, and then you do out of distribution questions on all sorts of other animals. So when we wrote the proposal for the semester-long program, we heavily relied on AlphaFold and her for motivation. So I'm actually extremely emotionally proud and happy to be able to have her give this talk. So take it away.
Wow, that's a little scary. Thank you. Thanks for the intro. And I was not aware of that, no matter how little or much of that is true. But anyway, it's a pleasure to be here. And I apologize in advance if it's a little chaotic, because I actually dug out slides I have retired to make this more suited to this, the topic of this workshop. And rather, I'm probably not going to go super in depth on any particular piece of work from my group, but my goal is to just give you a sense of what's happening in protein engineering now and some of the problems in machine learning that it's made us think about and some of the solutions that we've kind of proposed over the years. And feel free to interrupt me as it suits you. Okay, so I get till 12:50, I guess. Okay, so let me just give you an introduction to what's happening in protein engineering right now. But first to motivate it. So I'm guessing nobody in this room does anything with protein engineering. Anyone? Okay, so I can say anything I want to anybody except you. But as you may know, that this plays a very important role for therapeutics, like antibodies are proteins. There's a huge effort underway to use proteins such as innocuous viruses that we have a project with here on campus to deliver payloads that can be used for therapeutics. You all know about gene editing, in particular if you're anywhere near Berkeley or Boston. And the main enzyme that goes in and cuts the DNA, this is a protein that has been engineered tremendously. I should say these have all been engineered tremendously over the years. What you may not know is actually proteins are one of the workhorses for manufacturing antibiotics at production scale. Jay Kiesling here on campus is quite involved in that. You may also not know, luckily I don't see any of these plastic bottles, but like in many places, everyone's drinking these plastic bottles and people are trying to re-engineer the PETase enzyme to make it more suitable for that at industrial scale, which it's not. And the most abundant protein on the whole planet is called rubisco, and it's involved in carbon sequestration. And Dave Savage here on campus is trying to engineer this to have a better it has sort of a trade-off curve between several characteristics. And if we could get it off that Pareto frontier, it could stand to benefit the world quite a bit for environmental and agricultural reasons both. So this is like huge impact. And as you've heard me say, a lot of this has been engineered, but as you're going to hear, there's much to do still in protein engineering, even though there has been so much success. But like if we just step back as a very abstract thinker, like a machine learning person, say like what at the most basic level is protein engineering or protein design, it's being able to tell someone in the lab what sequence of amino acids to use that will then create the protein that will do what we want. And at the end of the, you're going to hear something about structure and about properties, but when we design something, we need to specify the sequence. That's the kind of fundamental problem. And we may need to hedge our bets and have kind of a lottery ticket set of sequences in order to have more probability. But of course, this scales really badly. So there's 20 amino acids in the alphabet if we stick to the natural ones. There's also some folks on campus here trying to generalize to non-canonical amino acids, which would make the space much bigger. But even with the natural set, you take a very small protein of length 50, and already the the number of combinations is close to the number of atoms in the universe. So it's clearly just a very unwieldy, massive space that we need to traverse through. And in general, as we're traversing through that space, we don't know any of the characteristics of the protein that we may desire. But of course, here we all are sitting, functioning with proteins. That's what's keeping us alive, hopefully. And so there's something has managed to design these proteins, and it is, of course, natural evolution, but it works at the timescale of millions of years. And so this is like a proof of principle that it can be done. And at the same time, it's obviously not practical for us to directly leverage anything going on there. Although, as you'll hear in short snippets, we do leverage some things that happen evolutionarily to help us in the modern era of machine learning-based design. And then the second strategy that actually humans have developed are these protein engineering strategies. And every year I got to add another Nobel Prize to this slide. So there's two main ways that people have been doing this. The first is computation-based. I say data-free in quotes because it's primarily biophysics-based, maybe instantiating a few parameters here and there with some small sets of data, but it's not using data in the manner of modern-day machine learning. I say almost rest in peace. This thing was conquering the world. In fact, it's why it got the Nobel Prize. And there are still corners. So despite what Mathieu said, is that some of these models can kind of generalize on some axes. On many axes, they can't generalize, actually, and there are still issues. And so some people are still using these, but I expect in the next few years that's going to stop, other than people who just really like them and don't want to give them up. And then the other one that got a Nobel Prize quite a few years ago, well, more years ago now, is wet lab directed evolution, which is basically like a greedy stochastic local search. You start somewhere in space, you induce some variation at random or somewhat informed if you can, but still stochastic. And then you screen for the top performers, you induce this variation again, and you keep going until you hit a local minimum. And the reason I'm here talking today is that somewhere around 2018, people started to ask which is actually the year I started here at UC Berkeley and started to think about these questions for proteins. My history is a long history in machine learning and molecular biology, but the proteins started for me just around this time. And so this is, machine learning is kind of infusing everything. There's a sort of culture that likes to pretend we're just going to have a model, we're going to design one protein, and it's going to be the protein we want. And people, depending on your niche and what your biases and what you want to conquer the world with, you may hide certain aspects. You may hide that you're doing directed evolution with your biophysics model or with your machine learning-based model. The reality is these are all helping to inform each other. Machine learning methods still usually, at the end of the day, you get a good candidate, you might need to do some directed evolution in the wet lab, and so on and so forth. In some way, I think they're all coalescing into like an approach that makes use of all of these. At the moment, it's not a single cohesive approach, but this is the trend. Okay, and so some question I get quite often is, well, like, why are you working on protein engineering? Didn't AlphaFold solve protein engineering? And so, like, to some people, they laugh because obviously the answer is no, but I get the question so much, I'd like to explain this near the beginning of my talk. So AlphaFold was a tremendous success, as perhaps evidenced by the Nobel Prize. But even before that, I think we all knew it was a huge success because it just was a phase transition in our ability to predict structure from sequence. But as I said, when we're trying to design proteins that have properties that we want for therapeutics or other reasons, we need to get the sequence, not the structure. So if anything, we would need to turn it on its head and say, I know the structure I need, you tell me the sequence. And in fact, my student Chloe on an internship at Facebook built one of these giant inverse folding models. She essentially inverted AlphaFold with massive compute and smart engineering. And so these models exist. There's quite a few of them now, and they work. They're not perfect, but you can get something out of them, and they're a key component in modern day machine learning based protein engineering. So one thing that's overlooked in almost every single paper, especially high profile paper on protein engineering, is the fundamental bottleneck challenge. Just whatever generative models and AlphaFold and all of this is, we need to know the link between the sequence and the function that we desire, or at least between the structure and the function that we desire. If we can figure out what structure to get, then we can make use of these inverse folding models. And fundamentally, we rarely know that very well. And if we know it, we usually know it in an interpolative sense, in that, like, you know, I have a protein that fluoresces green, I can create other proteins that fluoresce the similar intensity of green. But if I wanted to shine a third more brightly in what I would call extrapolative machine learning based design, then I'm in this problem of how do I get it to the models to generalize for design. And not all design is like that, but a lot of design is like that, be it for proteins, small molecules, materials, all kinds of things. So, yeah.
How well does AlphaFold do on non-natural proteins, like on design proteins?
I don't know the numbers, but I'll tell you where there are problems with AlphaFold in a couple of slides. They don't have that one on it. I suspect it's hit and miss, and that it's a question of like, well, it's not natural. Is it one sequence step away? Is it totally different fold? There's notions of all proteins that exist, even the synthetic ones, are often composed of Lego blocks that are known. So the question has to be more nuanced than that, and I don't know the answer, and I haven't sliced and diced it up correctly.
I think I was thinking
Yeah.
if I optimize, if I take AlphaFold as my mapping to like even to structure, like here, but taking aside this function issue, and I try to optimize the input, will I get to a place that like seems to me like, you know, AlphaFold will give me the structure I want, but that's because I went outside of its the space in which it perform as well, and I'll actually get a sequence.
I think there's a lot of people are questioning very much how well it works outside of the space it was trained on, but it's important to define on what axes you mean by out of distribution. And that itself is a complex topic. I guess that's maybe what I'm saying is defining out of distribution is not so simple there. Even though in machine learning, we just say it's a different distribution, but it's like exactly what does that mean is the key here. And I don't want to get too sidetracked, but I'm happy to talk about it after, yeah. So when I started working in this field, I got very disoriented because I'd read a new paper every week and they were saying they were solving all of protein engineering, but they were all doing different things. And so I kind of actually, my student Chloe, I think once kind of made this up of this set of prediction tasks versus a set of design tasks. You could call it a bit arbitrary, but if you go pick up a paper that's ostensibly doing, or that is doing protein engineering, it's solving one of these many tasks I'm going to show you on the 2 slides. And so the prediction tasks are things like, I give you a sequence, you tell me the function prediction, or I give you the sequence, you tell me the structure, I predict the structure, that's what AlphaFold does, or you give me the structure and I predict the function. And none of these are in the design direction. In the design direction, you want to be getting closer to sequence, and you'll notice none of these arrows are pointing to the sequence. So in contrast, you could call these problems design problems. And so here, ideally, you would like to just specify all the desired function properties that you want and get the sequence. And so this is super, super challenging. At the moment, people, there's a little bit that goes directly down this arrow, but usually it's a 2-step process in the most common ones where they specify a function and then they get the backbone structure. That's just the central atom of each amino acid, roughly speaking. And then from this sort of coarse grained structure, they then use an inverse folding model to get the amino acid sequence. And so these are kind of, people are working on all of these sub problems individually to make them better, as well as to string them together to do sort of a full pipeline of protein engineering. Okay, so what are some of the trends? These are going to just, oh, yeah.
Once you have the sequence, you can synthesize it. How?
Oh yeah, once I tell you a sequence, that's, well, it's fairly, it's easy, but it can cost money. So if you just need to build one sequence, like if you knew with 100% certainty, like that would be super feasible. But as you need to hedge your bets, because none of this works perfectly, the more sequences you need to create, the more expensive it gets. And this is another kind of hidden fact of machine learning based design that's quite interesting. I have some work I can't talk about that also doesn't fit here, but is how do you use machine learning with cost-effective, scalable synthesis. So the technology is there, but it can be extremely expensive. Normally, like for a short sequence, you just go to some company, and for about $10 for a sequence up to maybe length I can't remember, like maybe it's like a few hundred nucleotides, you get that one. But it could be $5 to $10 per sequence. And depending on your engineering, you may want to be making 1 million of them. And so something we do that I'm not going to talk about is sometimes we, instead of specifying the exact sequence, we use a cheaper synthesis method that allows us to specify a distribution with stochasticity that doesn't give us the same control, but gives us much, much larger scale for the same price. Yeah.
For sequence and structure, we have a nice formal way to write them down.
Yeah.
For function, how do you typically describe them?
So that would be different for different functions. So enzymes, there's usually like a catalytic efficiency in some like biophysical units. So brightness would be at a wavelength, a particular intensity. There's a lot of stuff is based on binding and binding. So you have like a binding affinity basically, or that you can get to more biophysical constants that control the like dynamics of how much it goes on binds, how much it goes off it binds, and things like that. But there's typically some biophysical ground truth measurement that one can do in small scale that's very costly. Most machine learning is using measurements that are not so precise, but that are much cheaper.
Have there been any effort to try to synthesize those different functions? It seems like that's a fracture that's fundamental to this, but it seems like they could also mutually inform one another in a useful way.
Which two things?
The functions.
Yes.
Right. Like you're measuring intensity or
oh yeah. So right. I think what you're saying is one, well, maybe you're saying two things. One is we may desire property criteria specifications on multiple properties. That's absolutely true. Usually we can barely get, like mostly we're focused on one, but in the therapeutic setting, like you need like solubility, non-toxicity, you need it not to be shut down by the immune system. Like in addition to doing the primary thing, you want to hit the target and not to hit the wrong target. So yeah. And sometimes if they're correlated, then indeed knowing one could help inform the other through some sort of multitask prediction. But I'd say that's not sort of, that's almost peripheral to this field because we have the main bucket is so hard. Yeah. So right, okay, so just some of the trends. These are not super interesting, but just to, so you know what people are talking about. There's 1 million people super excited about representation learning with what they call large language models. I call pan-protein models. They're training them on huge unsupervised sequences of proteins with the idea that they'll get to extract a representation that's useful for some supervised task for which they have very little data, and they hit all the buzzwords of transfer learning and and so really they're doing approximate density estimation here and extracting a representation from the density model. Another really big one are conditional generative models or generative models for sequences. They may be conditional generative models. And some of these are the same kinds of giant models that get used for the representation learning. Sometimes you can write a structure conditioned generative model for sequences. That's actually this inverse folding model that I told you about. But the idea here, again, is that design fundamentally means I give the sequence or the set of sequences to the lab. So can I get the sequences I want by conditioning on a structure or on some property or something like this? And then, yeah.
I think it's implicit here that the structure is kind of the sufficient statistic for what the function is going to be.
It's not actually. There's a lot of people think it is, and historically this has been said. So yeah, what I neglected to say somewhere, or should say coming forward, but like the things that are really within reach of being able to solve in protein engineering are those that are structure mediated functionality. So structure mediated functionality is primarily binding of two proteins. That's primarily just the actual structure. And again, we gloss over these things, and I'll mention in a slide, but really you actually need to know the dynamics. A lot of things undergo conformational change upon binding, and AlphaFold can't do like basically any of that stuff. So there's a lot of very important things. But so now what's an example of something where knowing the structure simply is not enough? Enzymes. And enzymes are a huge, huge part of protein engineering, super, super important. And there's fine-grained quantum detail at the active site that you cannot know. So you could have exactly the right structure and none of the right chemistry.
Not important. We're not going to talk about that. Okay. But this posterior assumes you have it prior over sequences. I mean, what should I think of as does that make sense, or what should I think of as the prior over sequences here?
Well, there is no prior. This is just
Because you're looking at, sorry.
This is just probability of sequence given some condition. Yeah. I mean,
Yeah, but if this probability of sequence giving some.
yeah, so when you do representation learning, you're implicitly talking about a prior over sequences and bringing it in. Yeah, so, but most of the field doesn't actually write it this way. But of course, everything has a statistical interpretation, and it'd be nice if people knew what they were doing. This is recorded. Okay, I haven't said any names. I have not said any names. Okay, let me keep going because this is the intro. Let me get to some of what we do. But I think this is a really big one. So there's a series of paper. John Ingraham is a super clean, brilliant scientist. He's at a company now, did his PhD at Harvard, and he did this beautiful, beautiful clean model on conditional general models for structure for the backbone, actually, again, just the central atom of each amino acid. The reason you don't specify all the atoms is because then you actually have to specify the amino acid. And if you want to use this two-step procedure where you first get sort of a coarse-grained backbone and then instantiate the sequence, first of all, it's cheaper because you don't have to model all the atoms, and it's basically what's within reach at the moment. And then David Baker's group has a similar one. But and so these are beautiful and elegant, and you like machine learning, you like to see the cat getting noisier and then less noisy. It's all fantastic. But the thing they're missing is that at the end of the day, you need something to inject to get this model to be conditional on the property you care about, either through guidance or bake it directly in, however you want to do it. And again, like if you're trying to build something with a property you've never seen, you're into this region of sort of extrapolative design. And again, like this, there's just people can't really do that. And so there's these, huge famous papers of, I took this enzyme and I re-engineered it. So what do they do usually? They take out the part that has all the important chemistry and they leave it fixed. And then they build out the rest of it and say, well, now I have a new version and it's smaller, but they can't actually really change the active site and the quantum properties. So that's why I put it in red, because on the one hand, like I sound like a skeptic. I think it's an exciting field, it's exciting times, but I think people bury the difficulties and highlight maybe more than what is warranted, or we don't know because there's not enough comparisons in the paper. And so if you're curious on like just a sort of primer style understanding of generative models in the space of proteins, you can check out this recent thing I did with my students who are now gone, Chloe and Clara. And then one that is really important, that's sort of the foundation of almost everything else, is how do I predict the function I care about from the sequence or from the backbone? And when we start an engineering campaign with our wet lab collaborators, so I work with a lot, we do some little pie in the sky machine learning stuff, but we work on the ground with wet lab collaborators over multi-year collaborations. And we usually need some understanding of the probability of the function we desire given the sequence. And usually we start with no labeled data at all. And we have to help in some statistical way design the first library. A library is like the first, I need to set the parameters of a distribution that is a stochastic mechanism to do scalable synthesis in the lab. And so what can I do in the absence of anything else? I can pick a uniform distribution over all positions in the protein, over all amino acids, and then it's hopeless because the space is just way too big for the scale of what I can measure, which itself can vary by order of magnitude. But so this is where we get into no-shot or zero-shot learning. And the zero-shot learning typically is where we do make use of evolution. We go grab all sequences that are through some heuristic algorithm known to be evolutionarily related. And we assume that anything that is in the set is enriched for the property we care about. And then we can build a density model on just those sequences. And we can use the density evaluation for test sequences to help rank what we think are better or worse for the property of the sequence that we specified to get all the evolutionarily related ones. So we need to know one sequence that has the property we care about to some extent. But so this is sort of a feel for how zero-shot learning comes in, and that it has its own very specific instantiation that's very domain-informed based on evolutionary information. And incidentally, evolutionary information is a really, really key part of AlphaFold. People like to think that AlphaFold is somehow getting at physics. It's provably not getting at physics because it cannot fold a protein for which it has no evolutionary information. At train time and at test time, the first thing AlphaFold does is, again, it goes and gets sequences that look like they're similar from the evolutionary in the large set of known proteins that are closely related. And if it doesn't find any, it will not work. So this is like a huge, huge bottleneck. But again, it's also incredible. Yeah.
Reference. Will it still return something? Or
Oh yeah. Well, so there's three different things. Let me get into the nuances. So you can always turn the crank. So you can give it, it can fail to retrieve anything and it will still give you a prediction. The other thing you can do is you can say, here's a protein. You could go get evolutionary related proteins, but I'm turning off that part through the API. How well does it work? And it can still work pretty well, actually. So it's not necessarily, well, it's a bit nuanced. So the question is, did it need it at train time? Does it need it at test time? But no matter what, it needs to have, at least theoretically, have that set, even if it's not using it. Okay, right. And so, right, this is actually what I'm saying is another trend. This one is a less popular trend because it's so hard. Like these, what are the gaps left by AlphaFold 2? So there's AlphaFold 3 now, which has just recently been released to the public. There was a big scandal because it was peer reviewed and published without accessibility to the code, or you could make like 15 calls a day or something like this. And so there's like people, was a huge outrage in the community, but they have just released it now for non-commercial use. But so, as I said, this example I was just giving what we are called orphan proteins, proteins with no or very few homologs, evolutionarily related proteins. Something that's getting better and that AlphaFold 3 is supposed to be better at is protein-protein binding or protein-DNA binding, protein-RNA. This one's really hard, but some of these are like progressing a little bit, but they don't work nearly as well as the core task of AlphaFold, which is structure prediction of one protein or multi-domain protein. But the really big kicker in which nobody can do anything is there's protein dynamics, which is like over time, how does it change? And we probably, for a lot of purposes, don't need to know exactly how it changes over time. But what we do need to know is the distribution over conformations, like according to some biophysics-based energy. We want to know what are the main conformations it takes on, and we may want to know which ones it can more easily exchange between, because these dynamics are very important as this protein functions in its role of binding to things and letting go and so forth. And therefore, it's equally important for protein engineering. And people are trying to run molecular dynamic simulations, which are kind of very approximate. They're biophysics-based energy functions in which you do some like kind of sampling to get trajectories. And so people have tried, for example, to inject this kind of information into AlphaFold the way it's trained. And so far, like there's no positive result. And so I think there's just a really interesting question out there that my students and I often ask each other, like, what do we have what we need now to get better at finding these conformations, or are we actually data bound at the moment, or are we simulation bound? Do we need more simulations? Do we need more precise simulations? Do we need more real data? I don't think that anybody knows, but people are trying to think of it along all of these lines, and that's like going to be the next frontier challenge that like hopefully one day there's like a Nobel Prize for that one. Okay, and so as you can see, I'm both excited and a little skeptical. And actually it's fun because I know there's two people in this room who disagree with some things. So I know Shafi does not. He never told me why you don't like this piece, but I know Shafi disagrees with this. But I've been frustrated by some of the things that are out there in terms of hyping up AI for science in general. And so if you're curious to see, and also I'd like to thank Ben. Oh, really? I thought you said you didn't agree with it. Okay, well, all right, then excellent. And I just want to, well, okay, there's something on one of my slides Ben won't like, but in the meantime, I will thank Ben because he was a fantastic help in editing this to be really clear. And so if you're just curious, like one thing I go over there is, why is it that AlphaFold, like DeepMind was very clever in the solution, but what DeepMind was most clever about was picking that problem. And what are the characteristics of that problem that made them pick it? And like, what other problems are there in science right now that have those same characteristics? And are there any at all? If you're curious, you can check out this paper. It also talks a bit about the difference in scale between modern day vision and NLP versus what some of the scale is in some of the scientific data problems. Okay, so that's my intro. What time did I have till again? 50. Okay, so I'm going to give you like a flavor of the kind of work that I've been. So as I said, I started at Berkeley in 2018, which is also when I started thinking in the space. And the very first slide I'm going to give you is like the first kind of project that we worked on. And I should say, I don't like to be, ironically, I don't like to be in very hyped up and crowded fields. And when I started this, I was like, oh, nobody's doing protein engineering and machine learning. But of course now, you know, Salesforce is doing it, Facebook is doing it, like it's all over, which is a double-edged sword. But I'm still enjoying it, but I try not to pick the problems that DeepMind and David Baker are working on. And so you'll get a flavor of what kind of problems I am working on and that I enjoy. So, okay, this is sort of the very first project we started to think about like this. So in general, in machine learning, we're like for a supervised model, you're usually giving it the training data, the inputs and some labels, and then fixing the knobs of the network through optimization, right? In design, one way to think about design, and what I'm going to say is true whether you think of it this way or you think of a generative model and guidance, whatever, the problem is nevertheless going to be here. This is just, I think, a cute analogy. So, well, many years ago in computer vision, people wanted to understand what a state-of-the-art vision neural network that takes an image and makes a prediction. Are you a banana? Yes, no. Are you a kiwi? Yes, no. Are you whatever? And what they wanted to say was like, I want to probe this thing. I don't want to train it. It's already trained, but I want to know what it thinks the quintessential banana is. So they put the banana node to 1, all the other banana nodes to 0. And then they ask what image is most banana-like. And that's basically a direct analogy to what we do in protein engineering. I say, make the catalytic efficiency, above this threshold. And then you tell me the protein, be it by sequence or structure or something like this. And so when the vision people do this, this is what happens. This, if you do this naively, you get the quintessential banana that the jumbled neural network thinks is this. And so on the one hand, it's plausible, right? It has the right curvature, it has the right colors. You go to 3D print it, then you're just getting abstract art. And if you're trying to do protein design, you're almost certainly just going to get some protein that doesn't even fold. Like that's kind of the lowest bar. Like, are you a possibly useful protein? If you have no structure, then you're not a useful protein. And the number, if you just pick an arbitrary protein sequence from the amino acid alphabet, I don't know the statistics, I should get it, but it's very low probability will even be a folding protein. And so this is kind of the main challenge. And like, of course, we all know this is, what's going on here is there's a very high dimensional parameter space, or even if it's not, you get this problem. And you're squeezing this network for all it's worth, right? You're putting the banana node to 1, and then you're saying, give me the most banana-like thing you've ever seen. But that's not kind of in the training data, right? In a sense, like you're pushing it outside the training data to get the most banana possible. And by analogy, this is also what's going to happen if we try to do what I call extrapolative design for protein engineering. And so there's this natural tension that arises when you do any kind of machine learning based design, again, be it for proteins, small molecules, materials, where you are often trying to extrapolate, but you know the model is not trustworthy. Of course, if you had a causal model, it would be, and of course, causality is impossible. No offense to anybody in the audience who works on causality. And so how do you, we can't solve this, but what's the best we can do? How should we think about it? And what tools can we bring to bear? And there's also what often comes in for us is this distinction between epistemic uncertainty and aleatoric uncertainty. Is it uncertainty from sensor noise, or is it uncertainty because it's like far away from everything I've ever seen? And that's when I'm extrapolating. I need to care about this epistemic uncertainty. Can I estimate it? If I can, how is it useful? And then something I thought was super boring, but got a little bit intrigued by, this is just a minor side story, is we don't often have much data compared to the common fields. And so in fact, what happens normally is people say, oh, convolutional filters, let me use those, or let me use this. Like they just port things directly over. But I think it's useful to sometimes stand back and say, are there things we could do that are specific to proteins or this particular science that are kind of generic, but might improve because the inductive bias is actually geared, it's very general, but geared to this domain. And so just, there's not much work in this, but a little flavor of this is, I don't know. Okay, it's funny. I have 4 citations. I don't know. I must have when I wrote this slide. Well, let me talk about just one of them very briefly. So the idea of inductive bias, like what's one example of what I might mean? So imagine I'm building estimating a function over sequences, so discrete state space on the bottom, and I'm predicting a real valued, let's say, binding affinity scalar at the top. And so one thing that I know, or if you're in this field, is the most general kind of statement you can make that's always going to be true, is the probability that think of that function instead of a neural network as just one large linear additive model where I get to have features that are arbitrarily complex, what we call like high order epistasis. They're just high order interaction features, and any function on a real value that I'm trying to estimate from this discrete state space, I can always write as this full linear additive model if I let the order of the interactions go all the way up, right? Kind of like a polynomial basis expansion. And however, we don't want to deal in that huge explicit combinatorial space, which is why we often use neural networks or other things. And so the idea is that in these functions, when it is on a protein sequence, then we know that the higher order interactions, which are kind of in a way like, think of it as like high frequency Fourier in a continuous space, that they're like less and less likely to truly be in the true causal function. And if they are there also, we may just not have the data to estimate them. So they're going to be in any case more unreliable. So the general inductive bias we want to put in is that the higher the order of the feature, the interaction, the less likely we want it to be in the model. And we want to encode that inductive bias into the neural network. And I'm not going to go into the details here. But the challenge is that I'm talking about a regularization on a currency which is in the full expanded combinatorial space, and I want to impose that regularization on a neural network which is described in a different currency, right? So I essentially need to take like the Walsh-Hadamard transform, which is like the Fourier transform, but for discrete state spaces. I need to basically take that of the neural network function and then regularize in that space. And that's a very expensive operation. And so in this paper, we kind of explore this. And I don't think it's necessarily like the way to go. I'm not selling this as you should definitely do this, but I think it's interesting to think in these directions that more people could be doing that. And the one that this is like my kind of niche thing that actually I didn't put much of in this talk. We have actually a published collaboration with David Schaefer's lab here for AAV therapeutics. But you know, we do in machine learning often think about, I want to design this one sequence that will do what I want, or because I'm uncertain, I want to design a set of sequences, which is then like a distribution defined by point masses. But as was brought up, or I answered to Misha, is that scalable synthesis at cost is at the moment very, very expensive. But no matter, and so we often replace this exact specification with a distribution that gives us less control, but attaches onto a process in the lab that's cheaper. And so I'm trading off my ability to fully control the exact sequence synthesis with my ability to kind of produce 1,000 times more sequences for the same price. And so when you start thinking in this way, you're basically designing distributions and you're hedging your bets on how diverse should they be versus what's their expected binding affinity for the thing I care about. And you end up with trade-offs and some questions related to active learning a little bit and so forth. So this is a very rich technical area as well. And so, yeah, so a couple of years ago, someone invited us to write a piece. I think it was on something else, but this is what we wrote it on. Basically, Clara, who's now at Genentech in one of the research units, has graduated, but she was still here. And we just basically put all our thoughts over all the years about like, what does it mean to try to design in this extrapolative sense? Like, are we assuming we can predict novelty of some kind? Like, what does novelty mean? And so it's a fairly general piece for machine learning based design, but it's instantiated in proteins. So if these topics are of interest to you and you don't mind the instantiation in protein space, you can check that out. Okay, so where am I? All right, so this is the very first paper I wrote with my student. I'm just trying to think how much time I have, and I'm trying to remember what I put in my talk so I can go over what I want to, but I can't remember anymore. Okay, let's just go with what's written. So yeah, this is an old paper that people like, but usually when I ask them about it or they ask me about it, I realize that they've misunderstood it. So I guess we wrote it very badly. But so what is this paper about? It's saying, imagine you have this fixed neural network for the property you care about, and you're trying to find some X that maximizes it. So you say, I want to find, I trained on a bunch of stuff for this binding affinity to that target. Now find me the one protein that has like twice what I ever saw in the training data, or the maximum, let's say. So max over this f of x of this function x. And so I can convert this. There's different literatures that have done this in different ways. It appears a little bit in RL. There's a superficial resemblance, but this is what we or other people, I don't remember anymore, start calling model-based optimization. So you convert the problem over the state space X to searching over the parameters of a generative model with parameters theta that generates X. And now you want to find the parameters of the generative model that give you the max expectation over this. So this is sort of a sleight of hand. And if you do this exactly as it's written, if this generative model has high enough capacity, it will just find the point mass setting of theta that gives the all of its mass to the 1x that maximizes it. So right now that's not necessarily useful, but it turns out that you can also add an entropy regularizer so it doesn't collapse. And now you have something that's very well suited to this thing I've been calling library design, which is a generative model. So long as you pick a generative model that matches your way you're synthesizing stuff in the lab that is cost effective. So if you connect the mechanistic cost-effective way to synthesize with the parameters of a generative model, now you have a way to do scalable synthesis and design a library. And so this is not something very many people think about, but it's like, it really is kind of involved in every engineering campaign that you do if you bother to think about it. And what's also nice is you've kind of converted a problem that doesn't have any statistical language to one that does. And so what we actually did in this first paper is make use of this sort of statistical language to bring in a prior over sequence space, kind of like a soft trust region that makes this procedure, well, this isn't a procedure, right? There's a loss function. Then there's a whole algorithm can kind of quickly, I don't know if I have it or not, but it's related to a whole bunch of stuff, something called estimation of distribution algorithms, which I like to think of as the modern day evolutionary algorithms without all the dirty associations with them. So instead of, old school, and people still use them, evolutionary algorithms, you define mutations and mating and whatever. Here, you just replace that with a generative model. You sample from a generative model, which in effect creates the mutations and so forth that you want. But I don't want to go into all of this. All that to say is there's a rich history of doing optimization in this kind of a way over the distribution. And there's, I think, potentially interesting connections there. And not super interesting, but basically what you end up with if you use that kind of technique, and if you're doing extrapolative design, you also need to consider the fact that you have a rare event sampling as you're doing this. So just a bunch of interesting stuff comes up. But basically you end up with an iterative algorithm that, in fact, we show in one of our papers, is a form of expectation maximization. And at the maximization step, you're always changing the parameter of the generative model. So it's sort of like a spotlight in your sequence space that you keep moving to the parts of the space for which the property you're looking for are expected to be higher. And so if you just do this naively, you're really just doing that model inversion, and you're really going to end up with that abstract banana because it assumes that the model is basically perfect. You're just doing optimization. And so we just had this sort of very simple idea of adding in a soft trust region. And the main, one thing is, how do you get the formalism to do that in this kind of algorithm? And then the harder question is like, how do you get that soft trust region? Like, where are you truly getting that information from? And so one thing you can always kind of do is just say, well, I know where I trained the model. Let me use the density of those training data as a sort of soft trust distribution. And there's always some catch there, right? Like, well, how much, if I use that in a too conservative a way, then I don't explore it all. And if I use it in not conservative enough way, then I go too far out. But at least you have some language now to think about it, and like maybe one parameter to play with a little bit. The other thing you can do, and implicitly people are doing this with the big representation learning type of problem, is take like all proteins known to exist and compute a density over them, and then assume that that density is, in a sense, like, how protein-like are you? What it's really probably doing is saying, how stable are you? I think implicitly, a density function over all known sequences in the database is like a stability prior or a folding prior. And so you can instead use that as a soft trust region. And I'm not going to bore you with all the details, but basically what happens in this EM-like algorithm is you get a weighting scheme that it downweights stuff that's kind of going too far out according to your soft trust region. And so, but something that got like more interesting was, so this was with my first student, David. And then Clara came along and she said, well, I wonder if, as you're doing this sort of EM-like algorithm, at every iteration, you're making a call to a predictive model to score the samples that you've just sampled from the generative model in order to move that sort of spotlight through the space. And so notably, we're not acquiring new data, but Clara, who's very clever, said, I wonder if, as we're moving through the space, we should change the predictive model, even though we're not collecting new data. And so in the end, in fact, the answer was yes. And again, I'm not going to go into the details, and she didn't set it up this way. She didn't think of it as domain shift. But in the end, what emerges from going through the math is something that looks a lot like domain shift. And so what's happening really is at each iteration of this model-based optimization, you're trying to update the parameters for the generative model. And in each iteration, you then sample from the current value of the generative model, and then you have to evaluate those under your predictive model. And as you progress, the samples are further and further from the training data of the predictive model, kind of by design, no pun intended. And so essentially what she proves is that every time you move through the space, even though you're not collecting new data, then under the assumption you don't have a causal model, which is 100% guaranteed, then you should reweight the training data, your original training data, so that it more closely matches the distribution of the current test data, which are the data that are getting further and further away from your training data. And just like in domain shift, there's problems that as you got far away, the variance is very high, the effective sample size goes down and so forth. So you kind of, if you combine it with the soft trust region, you modulate that problem. Nadi looks very, yeah.
Basically what you're saying is that I didn't have computational considerations here.
Yeah.
And the right thing for me to do is every time you give me a point to predict on that single point, I'll go and reweigh my training data and then use that in our
well, it's a bit tricky with a single point, because then, like, I think when you have a distribution of points, it's more sensible. Like, it's going to get well. I mean,
so what I'm trying to understand is this is completely nonsense. Right?
Yeah.
It's completely nonsensical
Yeah.
from a facial perspective, right? Because
Yeah.
I'm saying for every test point, I'm
Yeah.
going to retrain the entire thing. This
Yeah.
doesn't make any sense.
yeah,
But I'm trying to separate here the computational from the prediction aspect. In
Yeah.
terms of getting the best predictions, is this basically what you're advocating? What this is advocating that
yeah. I mean, if you believe, in a sense, if you believe the like Sugiyama papers of like important sample reweight your training data to match the test distribution, modulo issues with variance and effective sample size and so forth, like then in this sort of idealized setting, this is kind of the right thing to do. And exactly, yeah. And you're basically, I guess the main high level point here is when you're doing design, you're almost always doing an iterative optimization. And usually as you iterate, you're making a new theoretical library, which is a distribution of proteins, whose distribution is becoming further and further from the training one. And this is an example of that and what you might do to do a bit better.
I wonder if in your way of thinking there's an insight for evolutionary biology, because what evolution does at some level is also try and come up with proteins that are good at working.
Yeah.
And you can say maybe the components are there, and like in a cell, you will have all kinds of RNA and DNA species that are drawn from kind of the set of all possible proteins. And, so, and you have often types of immune systems that could be like, that could flag things that aren't possibly of the right kind. So I wonder if like one could even push that back to biology and say like biology should at least somewhat show a reflection of those optimization ideas.
I'm not sure I fully parse what you're saying, but let me try to just disentangle two aspects. Like one is, where can I trust the space? And how do I define a soft trust region? And how do I put that into whatever optimization I'm doing? And that's like what I was talking about. Now this is slightly switching gears, which is just saying like, should I ever update this predictive model I'm trying to invert, even though I'm not collecting new data?
No, that wasn't my point.
Okay.
My point is biology itself can be seen as a big protein design.
Yeah. Oh, yeah.
Design problem.
Yeah.
The question is, of the tricks that you're describing there, might we expect that biology is also using some of them?
Oh, I see. I very much doubt they're doing domain shift adaptation with important sampling by any lens you look at. I think it's too smart to do something so dumb. And as for the is it using some sort of soft trust region? No, it's not, right? It's like, it's just in evolution, in a sense, develops to work at a pace where it doesn't make too risky a bet, right? Like evolution works at a pace, it makes changes where many changes fail, but enough work that you get evolution. So it's almost like through evolution figured out how much it can change at any given time. And different species, depending on the robustness of their genome, will have higher or lower evolutionary rates and so forth for that reason. Yeah.
I mean, also with evolution, it only has the actual, like the objective function it's optimizing is the real fitness objective function, not some kind of like wacky neural net-based approximation that you're optimizing.
Yeah. Also.
So in that case, we have to even work.
Okay. I think I should like be done a minute ago, roughly. So I was going to tell you about some other projects that had some of these themes, but I'm in the interest of time, maybe I'll just like have 5 seconds on a few of them, just in case you're curious. In this paper, the idea was to use epistemic uncertainty to the extent we can estimate it with Bayesian neural networks to combine information in a kind of rigorous, coherent way with biophysics-based prior information so that we can get the best of the both worlds as we're extrapolating. Like we know machine learning models are going to be good near the training data. As we back off, biophysics models are probably going to be better. How do we gracefully go from one to the other? This is the one that Ben will hate. This is just, but I'll just very quickly, if you've heard of conformal prediction, it's this like magic sauce where you get finite sample guarantees, except you don't get it on the actual thing you care about, usually the prediction marginal distribution. You get it in expectation over all training test data. And the fundamental idea is you assume exchangeability of the training and test data. And because basically you're doing like a permutation test and then counting statistics. And so if you have test data that's different, you can put in like a resampling for the kind of permutation type test that accounts for that. And what Clara did was kind of define and correct for a more complicated difference, which is not just the test set being different from the train set, but there's an actual dependency between them because you're designing the test set as you do design. So just a bit of a teaser there, not going to go through it. That I mentioned. And then, okay, just one, if you're interested in like, where is generalization coming in? I'm just gonna say one more thing and then I'm done. Okay, so I had a whole thing because I really just threw a bunch of stuff in here today. I had no idea how long it would take, but let me just highlight that one thing people, is very common. Diffusion models and flow-based models are all over science now, for good or for bad, maybe a bit of both. And it's too bad you don't get to see that, but okay. But the main point I want to make is like, where is there something where distribution shift and generalization are really coming in? It's when you do guidance. So when you do guidance with diffusion, you have this predictive model that is adding signal with this sort of Langevin dynamics to the gradient of the unconditional model. And there's actually usually a huge distribution mismatch of where you're trying to go, what it was trained on, like kind of all the same problems arise here. And people aren't spending that much time looking at them. I think because for many problems, they're kind of roughly okay. But I think in the sciences, we're kind of roughly not okay, mostly. And so there's like, I've seen a tiny bit of work here. And the question is like, is all the work here just make this model as robust and generalizable and extrapolative as possible? Or is there something specific about diffusion and guidance where I should rethink the problem in that context? Food for thought. And so my students did a paper on how to do guidance in discrete space, which is where these slides are from. And I'm just going to stop. So we have a bunch of collaborations. I don't just make up weird random machine learning stuff. And so I'll just stop there because I'm already way over time and leave it there. I don't know if we have time for questions.
Because all the talks have gone behind it, people need to get lunch. Super unfortunately, I'll say.
There were lots of questions. There were lots of questions. So
everybody can come up.
All right.
Enjoy your lunch. We're back in 2 minutes.